Hi everybody, good afternoon. Welcome back to session 3 of the workshop. I hope you've all had a chance to dip into that resource pack that you were sent, if you were sent one. I've just made a little dip into the Wicker bar and it was extremely nice, recommend that. So we've got 3 talks this afternoon from Joe Carpenter, Dean Anderson and Dave Kelly, but before we get to that, there's a chance to have questions for the speakers from the previous session. So they are duly assembled here, Adrian, Joe and Oscar. And I've got some of the questions that weren't answered. So I just thought I would run through some of those. So and I'll run them together for each person. So Adrian, maybe if we start with you. If someone says if epiphytes could affect rat density, how will you measure rat density? Is it known how often rats in high density epiphyte forests come to the ground in order to be tracked?
Yeah, so I was just talking to John about that before, and I think it's probably best if I just throw that to John, given that his expertise in this area.
And I just said to Adrian that people have asked me this all my ratty life, and I don't myself believe that there are rats that just stay up trees. I think if it were true, then results that we measure up trees like bird nesting success would be at odds with indices we take on the ground, and that's not the case. And also when we've done rat radio tracking, those rats move really freely from up in trees to down on the grounds. But no one has actually addressed that question with precise research, but I think they move freely between the two. Adrian, do the acoustic recorders give you a complete list of the birds present, and how long were they run for?
So the answer to that is no. There are some birds that are quite hard to detect. For instance, keriru, you know, like they're generally fairly quiet, and sometimes if they're really, really close, you might get the little cooing, but generally not. So no, they don't pick up everything. How long they'll run for? We collected 3 days worth of data, but only 1 day has been analysed.
Okay. Here's a question that interests me. I was wondering if rat vocalizations ever get picked up on the acoustic recorders, and if so, could this be used to determine rat occupancy to aid in eradications?
We haven't got any recorded in the data set, so I'm not sure whether I'd have to go and have a talk to some of the people who actually analyze those data, because they weren't specifically asked to record rats if they heard them. So I'll get back to you on that one.
Yeah, I've heard some of the questions about wetter vocalizations as well. You know, that I think at night maybe there might be less clutter on the acoustic recorders, and it's possible to pick some of these things up with a bit more clarity than in the daytime. Adrian, if fruit hangs on a tree across more than one sampling session, is it counted again? You know, is the long fruiting season due to fruit keeping better up in the fridge?
So yes, they are recorded, but I mean, the idea here is to understand how long that resource is around in the environment, and so you know that's kind of part of it. If fruit are there for a long time, then that's part of the story. But we will be analysing those data in a bit more detail to look at how much of that is due to kind of particular species versus, you know, interspecific variation and all the rest of it.
Okay. Joe, if we turn to you, is decreased quantity or quality of litter influenced by water availability? Or maybe they mean, does decreased quantity or quality of litter influence water availability? I don't know, you might have to work your way through that question.
Yeah, I think the latter way you worded it.
Okay.
Yeah, it would definitely affect water availability, and obviously deer consume an enormous amount of leaf litter, so ungulates could be having a really large effect on the amount of litter that's on the forest floor. And Tua Whenua elders have really noticed that there used to be these lush crops of fruit that were just, it was squishy underfoot, but that doesn't happen anymore. It's now dry and crackly underfoot. So I guess there's definitely been changes in that substrate, and I'm sure that affects water availability too.
Just as an auxiliary question, has soil moisture been measured in relation to ungulate movement, do you know? you know, like exclosures and things? Have people published about soil moisture inside and outside those exclosures?
Maybe David Wardle might have done something along those lines. I'm not entirely sure.
Okay. Someone says, what a cool study. They were talking about your BACI experiment, I think. How do you assess drought from stable isotopes?
Well, a tree, its leaves have pores called stomata, and these pores take in carbon dioxide and they let out water. But obviously if it's hot and dry and the tree needs to hold on to more water, it closes its little pores. And what that means is it ends up getting more carbon dioxide, or carbon-13 specifically, in the leaf. So it enriches the carbon ratio, and that gets laid down in the tree rings, and we can pick up on that enriched ratio based on the tree trying to save itself from drying out.
I'd just add in there too the oxygen stable isotopes are quite important for determining drought stress as well.
Okay, thanks, Adrian. Here's a slightly long question. Joe, has any work been done on the toxin response of native trees on the possible decline of native bird reproductive fitness? An example is the rongoa response in Kawakawa, whereby it increases in correlation to caterpillar damage. So too might other tree species respond to forest mammalian browse like tawa, karaka, etc., as it tries to inhibit imposed damage on heavily impacted individuals.
So the way I read that question is that with mammalian browsers, trees might be putting more toxic compounds in their structures, and that might be affecting birds. I don't think that is likely of fruits. And when we're talking about fruits and kiriru and tui and koromako being able to eat fruits, I don't think the pulp is likely to be becoming more toxic. Maybe the seeds might be, if they are getting destroyed by mammalian pests, but the whole point of these birds obviously is that they consume the whole fruit and they pass the seed intact and aren't affected, for example, by karaka seeds, which are highly toxic. You know, kiriru still eat the pulp. So I don't think that's likely from the fruit perspective. Potentially for a bird like kiriru or kōkako that eat foliage, different changes in secondary compounds might be occurring, but I doubt the trees have the ability to change those compounds rapidly enough in a way that would affect birds, because they just haven't had enough time when you consider their incredibly long life cycles.
Sounds like it would be something interesting to look at. Oscar, if we can turn to you from Dave Kelly, remarkable collapse of seed production in Otamatuna since 2014. Any idea of local factors that could cause it? Has it been very dry there for the last 6 years?
Yeah, good observation, Dave. Nothing jumps out in particular. I mean, there's a few years that are really dry and a few years that are quite warm, but it doesn't seem to be out of the ordinary looking at, say, the 5 years prior when fruiting was all right. So I think there might just be some other factor that the model's not capturing. And also we don't know what, you know, kind of possum densities might be in that area, which could also impact how many fruit actually reach the traps. So could be something like that.
Possum densities were measured for many years, of course, at Otamatuna being a mainland island and ongoing in relation to kōkako. So maybe there are data there, Oscar.
Oh, cool. Okay. Look at
that. This was for me, actually. The largest fruits I remember in your diagram were for Mongatautri and Rotakere. Is that an accident that they are pest fence sanctuaries?
Possibly yes, possibly no. When I looked at how fruit size changed like across latitude, it does kind of increase moving from south to north kind of, and the peak around those sites seems to follow that trend quite well. So it's not like they stand out as anomalous compared to the ones that are non-fenced and at a similar latitude along that kind of temperature gradient. But that being said, can't rule out the fact that absence of pests has some positive influence on fruit size too.
And from Josh Kemp, probably our last question. Vertebrates store resources by laying in fat. How do trees store resources?
I think might hand this one over to Adrian. So you probably know a lot more about that than I do.
Yeah, so there's obviously storage of sugars as starch. In terms of how much they store sugars for things like fruiting and flowering and stuff, suggests that it's not actually that long. You know, like a lot of the energy that goes into producing fruits and stuff and trees comes from the current year's productivity. Nitrogen's a bit different, and there's a bit of evidence that previous years' storage of nitrogen will contribute to crop size in the following year. And nitrogen gets stored in all parts of plants tissue, like particularly in the leaves, but also in stems and roots and all the rest of it. So yeah.
Great. So thanks, Joe and Oscar and Adrian. So this pattern of being able to sort of retro ask questions or to catch up with questions for the previous session at the beginning of another one, that will continue through the rest of the workshop. So